one of the things I'm going to talk about, I look at operant learning. When an animal basically performs a particular behavior for whatever reasons, and then if they are rewarded by that, then they're more likely to perform that behavior again. They're reinforced. And that reward, I started out studying drugs of addiction like cocaine or heroin. I love doing, playing with those drugs. But, yeah, that always gets that. But it quickly became clear that I think what I'm looking at is more of a fundamental mechanism of learning and operant learning. And then drugs are just another reward. So some of the rewards I'm going to be talking about are cocaine or heroin. And then in some experiments, I'm going to be talking about why an animal would press a lever for food reward, because it is an easier experiment to do. Okay. I always say learning plays a really big role in addiction. And I don't think we've really been looking at the mechanisms directly for it until just recently. Now, learned associations are formed during operant learning between the drug reinforcer, or food, and the other cues in the animal's environment. These are stimuli. And they could be I'm going to use this. They can be the instruments used to take the drug, or they could be the environment where drug is taken, or it could be your party friends. And now everybody seems to recognize this whenever I do this. Certain friends, you instantly see them and they go, oh, there's probably a party involved somewhere here. And then most of your friends, hopefully not all of them are your party friends, but there's the reason these become cues that can induce you to want and crave that drug, or sometimes food, more. And this could lead to relapse. And because it can lead to relapse, it's important to understand the mechanism so that we might someday be able to manipulate that. Now, the reason I like showing some of these examples is that they are complex sets of highly specific stimuli and that are encoded as high-resolution information. For instance, look at the person. They have the same general shape, one person to the next, but there's very specific aspects. It's not just one blob versus another blob. You need to have high-resolution information to do that. Unfortunately, most of what we've been looking at in addiction are more generalized changes in the brain that are incapable of encoding this high-resolution information. So when people look at kinds of changes in the brain, we call them neuroadaptations, and they're trying to understand certain aspects of learning and memory. Generally what they do is they will look for changes that happen in whole brain areas, or perhaps per specific cell types. But you can rapidly see that this is a very low resolution mechanism for encoding anything. This is like a 1-pixel camera that gets brighter and dimmer. It cannot encode the kind of information that we're interested in. So, and not only that, if that's how memories would be stored in the first place, then how do you memorize everything else that you ever memorize in your life? There has to be some other mechanism for encoding this kind of information and allowing you to discriminate between different memories. I just told you that. Basically, we'd like to, there's an idea, it started a long time ago, actually way back in the Nestler days. But Eric was saying, no, learning's got nothing to do with this. So I never actually explored this. But later on, I started hanging out with a bunch of behavior people, Roy Wise being one of them, and actually Yavin Shaham, which my colleague, he's actually did his undergrad at Hebrew University. And they got me interested in behavior. I was a molecular boy, and I was thinking, oh my gosh, this behavior stuff is fluffy. I don't want to deal with it. But then I really got interested in it. And the more you get to know something, the more interesting the questions get. And then you start seeing how you could merge your molecular work with behavior work. And one of the first things I realized is that behavior people don't really care about the increases or decreases in activity. They're more interested in why an animal or a person responds to one cue and not the other cue. And this is this information aspect. And this is what the brain does. It processes information. It doesn't just increase or decrease activity. How can this happen? So that high-resolution information Think of it this way, that a memory can be encoded inside a given brain area by activating a specific pattern in the brain, a specific pattern of neurons. In this case, I'm showing you in the red dots just some cartoon example that we have found recently anyways, that about 1% of the neurons react to a specific set of cues whenever there's a recall of a memory. And if that's that 1% of neurons, and you get one memory for one specific, what we call ensemble. This specific pattern is called an ensemble. If we get that one pattern for one memory, and there's only 1% that are active, and you've got hundreds of thousands, if not millions of neurons in any given brain area, you can imagine there's a near infinite number of patterns for the near infinite possible number of memories that you could have. So this is why we think of encoding high-resolution information in this manner. The other aspect is, if it's a different memory, different pattern. And this allows you to encode different memories using the same brain area. And there can be some overlap between what we call ensembles. So you can have all these memories that we're talking about. And this idea was started, well, actually written up back in 1949 in a book called Organization of Behavior. I don't know if that's taken up in psychology books here. I've got a shake of the head there. But this is an extraordinary book, really. When you think back in 1949, the kind of information that they had, or he had back then, it was extraordinary that he took these 3 or 4 really different ideas and then put them all together. It's really incredible, right? You know, but you should check it out. Chapters 1 through 5 of Organization of Behavior. Anyways, since then, people have been looking at using in vivo electrophysiology, you know, sticking an electrode into the brain of a rat or some animal, and trying to correlate that with behavior. And this has been these correlations for a very long time. But what we wanted to do, what I wanted to do, was manipulate these patterns so we can show a causal role for this pattern to demonstrate that what we're looking at is actually mediating the memory recall. And also, once you've identified those neurons, I think for the first time, then you could start looking at the molecular and cellular mechanisms that are truly underlying learning. Because if you just mush up the entire brain area, you are going to lose the kind of mechanisms. You will no longer be able to see the mechanisms that are really and truly mediating learning. So I'll tell you a few stories of how we got to the point where we are. Part behavior, one part is about molecular alterations, and the last part is about electrophysiological alterations that happen only in the ensembles that I'm looking at. And I'm defining them as the ensembles that induce a protein called Foss, and I'll get to that. I'm going to start. The first story is looking at an area of the brain called the Nucleus Accumbens. And this brain area basically takes information from a number of different sources, including the thalamus, prefrontal cortex, hippocampus, and amygdala. It takes this excitatory information and integrates it. Those neurons that receive the most excitatory, it's my first language and I still have problems. But when it integrates the most excitatory input, basically only those are going to be activated enough and persistently to induce the Foss protein that I'm going to talk about. But it's not just any random set of neurons. They are responding to a specific set of environmental stimuli, a specific set of drug-related stimuli, or even food-related stimuli. And this information selects that pattern within the nucleus accumbens for being active. And then cocaine basically amplifies that pattern. So somebody had pointed out that most of the pictures of my postdocs have somebody holding beer, and it's true. This particular picture is Fabio and Rodrigo, and they're wearing their Brazilian team shirts. And but basically, I'm going to start with a particular behavior that's commonly described in our addiction group. And this is something that was started by actually Yavin Shaham, my colleague at National Institute on Drug Abuse. I'll go through it. Basically, animals are trained to press a lever to receive, in this case, cocaine. And it's a good one. And But it's a specific set of environmental conditions. And we're going to call that environmental conditions context A. And then, after they've learned to press the lever for that particular, there we go, that particular drug, then they're placed in a different environment, a different set of stimuli. And in this area, they no longer get drug. So now they know that in context A, they get drug. In context B, they don't get drug. So on test day, you place them in the extinction environment, we call it, context B, where they don't get drug. Or you could put the other animals in context A, where they're used to getting the drug, in this case, cocaine. And basically, the animals will train during the first bunch of days to press the lever for cocaine. You put the dose of cocaine in half, and they'll press twice as hard to get the same amount of cocaine. And then they'll extinguish in context B. That is, they'll decrease the responding on the lever because they're not getting drug anymore. But on test day, you can see when they're placed back in the B environment, the context B environment, they don't press again because they're not expecting to get a reward. But if they're placed in the drug-paired environment, they do get the drug reward. They're expecting to get the drug reward even though they're not getting it on test day. And they'll press quite a lot for that. And that's called context-induced reinstatement. Okay. This is the behavior component. Now, inside the brains of these rats, we wanted to look which neurons were particularly active. And to see that, we use a marker. Many of you probably already know about it. But this is the Foss protein neural activity marker, which I've been studying for probably longer than you've been alive. But some of you, anyways. But basically, it's a protein inside the nuclei, and they're labeled brown when you label them in this manner. In this area, this is the nucleus accumbens, the shell, and the core subregions. But what you notice is that they're spread out, they're sparsely spread out patterns of Foss positive nuclei. And what you should know about Foss, there's thousands of papers on Foss. And a lot of it is dogma, a lot of it really is from studies where people have looked at cell culture or slice or anesthetized brains of animals. And much of that really is not related to how Foss is induced in a behaving animal. In a behaving animal, much of Foss is driven by neurotransmitters that activate the neuron, in this case glutamate, and sometimes acetylcholine can do it. And this neural activation opens up calcium channels so that calcium can rush in. If there is enough calcium rushing in and it's persistent enough, it can overwhelm the cell's ability to rapidly buffer calcium, which is what normally happens. Now, if there's enough calcium, it will activate the MAP kinase pathway that you see. I call it ERK MAP kinase here. And that will activate the CFOS promoter. And once you activate that gene, you get Fossa expression. Anyways, that's the explanation. And I use this Foss promoter in many of my procedures in the transgenic animals that I use. Let's get back to that behavior. When we look at the Foss expression using immunohistochemistry, what we see is the following. That in the extinction environment, where they don't get drug, there's very low levels of Foss expression. When we look in basically the drug-paired environment, context A, we see an increase in the amount of Foss expression that we get. Now, there was a control that I should have mentioned back here, that some of the animals on test day were placed in a novel environment that they had never seen before. And the reason for doing this is explained here, is that when you do that, there's a large expression of Foss. Okay, and this becomes really critical to remember for the next part of the experiment. There's a great deal of Foss. And I want you to memorize. Extinction is does not induce much Foss in this brain area. Drug-paired context more, and then novel context induces the most. And then the other thing we can do is we can label the Foss-positive nuclei, in this case green, and all the other neurons around them red using an antibody against nu-N, which is found in all neurons. And we find generally in two-dimensional slices that it's usually about 3 to 5% of neurons. But when you calculate this for a volume, it's generally less than 1% of neurons induce Foss. And if you have a very, very well-trained animal for a particular behavior, you find that very little, even lower numbers of Foss are actually turned on. But you can still show that very low numbers of Foss are mediating behavior. So we have that sparse expression of Foss, of a very low number of neurons. Now, to demonstrate that these Foss-expressing neurons are actually doing and mediating the behavior, we have to have some way of only manipulating those neurons, being able to either get rid of or inactivate those particular neurons. To do that, we use a transgenic rat that has the C-Foss promoter. It's an additional transgene. It has the C-Foss promoter that's turned on by neural activity, and when it does, it activates the LAX-Z gene, which makes the beta-galactosidase protein. And stop me if A, I'm not explaining something well enough, or on the other hand, if I'm explaining it too much. I'm just aiming for a certain place. Basically, this beta-galactosidase is found in the same set of neurons as the Foss neurons. Basically, wherever there is activation of a neuron to get Foss, there's beta-galactosidase. Now, the other part of the story is a drug, or a prodrug, called Donna-2. A prodrug meaning it is inactive in its beginning state. It needs to be activated. And Donna-2 is catalyzed by beta-galactosidase found only in these strongly activated neurons. And it's turned into what we call Donna-ribosome, which has been around forever in the cancer world for decades. And when it is turned on, created in these activated neurons, it basically kills those cells through apoptotic method. So that's the plan. Now let's get back to that context-induced reinstatement. Now, in these transgenic animals, we have context A. They're trained to press the lever for that particular, for cocaine. And then their behavior is extinguished in that separate environment, context B. And then we have what was called induction day. This shows up a lot. What you're trying to do on induction day is reactivate that same pattern of neurons, that same ensemble that is turned on during a recall of that memory. In this case, we placed them in context A. It's hypothetically, or actually what happens is that a particular pattern of neurons induces Foss and beta-galactosidase. And then 90 minutes later after we've done that, when there's maximal levels of that beta-galactosidase, we inject Donna-2 or we inject a vehicle control, in this case into the Nucleus Accumbens shell. And then we wait 3 days for killing off those neurons. What we see on test day is that if you ablate the neurons that are turned on in context A and then test later on context A, there is decreased lever pressing in the presence of those stimuli that make up context A. Okay, this demonstrates that those Foss-expressing neurons are actually mediating the behavior that you're interested in. Now, I could have shown you, I could have injected any nasty drug and got that effect into the Nucleus Accumbens shell. The main thing that makes anything interesting, I know it hurts to hear it, but is controls. And in this case, I used that novel context C control that I mentioned before. And the reason I used this one is because it produces a lot of Foss, a lot of beta-galactosidase. And the idea here is if it activates a different pattern and induces beta-galactosidase in a different pattern of neurons, and it kills those cells off using Donna-2, then the initial drug-paired ensemble should remain. That's the idea. And then so that when you test in context A later on, there should not be any effect. Lo and behold, thankfully, that is what we found, that it did not have an effect. If anything, slight increase. The reason I like using this novel context C is it induces a lot more Foss, a lot more beta-galactosidase than any of the other conditions. This basically hammers home the point. It's not the amount of Foss, it's not the number of neurons that are being activated, it is who is being activated. It's that pattern. I'm trying to get away from all this stuff, all this neuroadaptations about something increasing or decreasing is necessarily mediating. It's the pattern that matters. And trying to get away from what I call soup theory is that it's not a matter of adding a particular protein or mRNA or decreasing it. It's what the brain does. It processes information, and it has to have some way of doing that. And this is the way I can think of it. Oh yes, of course, naturally the beta-galactosidase that was induced on that test day is decreased when you kill off the context A-related neurons, but not when you killed off the novel context neurons. Skip ahead. Okay, that's one story. That's basically showing you what happens when you take out one particular ensemble. Well, then people might say, well, it's not that interesting because we still know it's that brain areas that's involved. What makes it more interesting is the following, and there's numbers of examples of this now. I'm looking at a part of the brain called the infralimbic cortex in the prefrontal cortex area. I'm looking at it with using food self-administration, not cocaine, but food self-administration in this case. Now, the idea going into this experiment is that self-administration memories and the extinction memories are separate. And this has been known since, obviously, people have pointed this out before, the days of Pavlov. But that being the case, people still didn't know, are the same set of neurons just changing and reacting to the same stimuli in different ways, or is it different sets of neurons? So the idea being is that an animal will press a lever, self-administration, and they will gradually increase that to get food. And we think of this as being due to a set of memories, or a self-administration memory. Consider it a set. But if you take away that food reward in this case, then you induce what's called an extinction memory. That's the idea. And it's thought to be due to a different mechanism, or a separate mechanism that's involved in storing that extinction memory. And we know this because the self-administration memory has not gone away. You can still reactivate it later by giving them drug-paired cues or context or even a little bit of the drug itself to remind the animal. So then we're looking at the infralimbic cortex. Some people say it's responsible for increasing lever pressing for a drug or food. Some people say it goes the other way. There's this mixed effect in the infralimbic cortex. So we think that both of those things are stored in the same brain area. So this following experiment, I'm going to go through it step by step because it's not always the easiest, is to look at the separation of self-administration and extinction memories. In this first experiment, or the first group that we're going to call the zero extinction session group, animals learn to press a lever to get food, palatable food, 1 hour each day for 7 days. And then, just to allow them to stay accustomed to that box, they're kept in those boxes day after day. Yes, we feed them, we give them water, but they don't have the levers present. So that way they're not able to extinguish, like press the lever and not get something. They don't extinguish that behavior. And then on test day, they get a short exposure to the levers present, and they're allowed to press it for a little while. And this is not enough to extinguish their behavior under our conditions, but this is to reactivate the self-administration memory that they learned over here. But the reason for doing this is that we start by examining Fossa expression. At the end of this, we basically perfuse the animals with paraformaldehyde and process them for Foss. In this separate set of animals, this is the 2-day extinction animals. Same training, 7 days each day, or 1 hour each day for 7 days. They're kept in the no levers box for 5 days, but for 2 days, 2 1-hour sessions. They're presented with the levers and allowed to press to get, basically they don't get anything. There's the production of an extinction memory under these conditions. And then we test later for Fossa expression. On the last group, it's the 7 days extinction group. Same, they're trained the same, 7 1-hour daily sessions. They're presented 1 hour each day for 7 days to extinguish that behavior. And then they're allowed to, you test basically how much they're willing to test on that last day. And all of these days we're looking for Fossa expression. And you can see, just to start off with the behavior, we're looking at 0 days, 2 days, and 7 days. The idea being trying to detect where we can separate the two memories. Animals will increase pressing for that lever during training, and when they extinguish that behavior, they extinguish it rapidly. And you can see this over here, that on when they did not undergo any extinction experience, there's a lot more, they press the lever a lot more. But then when they've gone through the experience of pressing a lever and not getting anything, they don't press as much on day 2 and day 7. So when we look at Foss expression in these animals, we divide each of the groups. The control group, half of the group is kept in the home cage, not taken anywhere, and we perfuse them looking for Foss expression. And what you'll find is that there's an induction of Foss expression on day 0, 2, and 7 relative to the home cage animals. But although there's an increase when you're reactivating that self-administration memory, there's actually more of an increase in Foss expression after the experience of 2 days of extinction, and still at 7 days of extinction. So it's all fine and dandy, but we want to use that Donna 2 experiment or procedure that I'd shown you before to be able to separate the two. Can we activate the self-administration memory and kill that? Can we activate the extinction memory and kill off the ensemble that's underlying that? And what you see here in the experiment, I always believe in explaining the experiment very clearly or else I lose people. But here's the food self-administration experiment. That same no levers exposure for 7 days. And then on that special day where you're trying to reactivate that memory, they're exposed for 15 minutes, very short span of time, to the levers, but they don't get that food reward. And then when beta-galactosidase is maximal in these transgenic animals, you try to kill off those particular neurons that are reactivated at that time, trying to kill off the self-administration ensemble that didn't have any extinction, or the extinction ensemble that had 2 days worth of extinction, as you see here. And then you test what happens if you kill off that self-administration ensemble versus the extinction ensemble. You see the lever pressing increases during training and decreases during the initial extinction. But on test day, when you look at the animals that did not get extinction, and that is where we're reactivating the self-administration ensemble, what you see is that Donna 2 will decrease lever pressing, is what you would expect anyways if you're ablating or killing off the neurons that are responsible for that particular self-administration memory. Now, in the very same brain area where we injected Donna 2, if the animals were exposed to 2 days of extinction and then later on you try to kill off the neurons when they were reactivated, what you get on test day is an increase in pressing for that lever. That's removing that extinction ensemble increases their lever pressing. Okay. It did not have to work that way, but it seems in this case that the extinction ensemble in this particular brain area is somehow actively repressing the self-administration ensemble. So the interesting thing about this is that you have a self-administration ensemble and you have an extinction ensemble in the same brain area. And there's, what we suspect is that they're intermingling right now. This is not something that you could get with other procedures, not something that you would get with other methods that existed before. But this raises the question, what does it mean when you use techniques that inactivate whole brain areas, like in this case, mucimal baclofen? These are GABA agonists. That is, they activate inhibitory receptors that keep the neurons quiet. In this case, what does it mean when you're inactivating something that makes behavior go up and at the same time inactivating neurons that are responsible for making behavior go down? What's the net effect? It's kind of a mishmash. You really can't predict. But that's basically what we've been dealing with for a very long time. So just to give you an example, this is something that Brandon Warren, the postdoc who did all of this extinction work in my lab. I'm showing you the behavior that I just showed again, and take a look at the extinction ensemble inactivation experiment here. Had we inactivated that same brain area using optogenetic methods like halorhodopsin mediated or baclofen mucimal, you notice that you do not get this increase in lever pressing when you inactivate the neurons in that particular brain area in those animals. This is something that only pops up when you're inactivating specific neurons. And indeed, when you're decreasing the self-administration ensemble, it's highly variable and never ends up quite like what you get when you inactivate the specific neurons that are mediating it. So, and there's many examples. This is just a list of examples. We've used this procedure quite a bit. I mean, going back to 2009, really. And more and more people are starting to use it now. It's actually a procedure or an idea that has taken a long while, a lot longer than it should have been for more people to get into, unfortunately. Because quite often the people who get into looking at these kind of things are molecular people or cellular people. And to explain to them quite often, because I was one of them, to explain to them that you're not interested in what makes some behavior go up or down. What you're interested in is why the animal responds to one cue and not the other cue. That doesn't concern them. I don't think it really crossed their mind. And that's, at least especially in the United States, I find it's much easier to explain all of this here in Europe and Israel, because the neuroadaptations hypothesis is so dominant in the United States right now. It takes a bit of time to get through. So I'm trying to figure out how long I'm trying to time everything. Okay. Now, that's all fine and dandy, but really, I am interested. I mean, even though I get into behavior, I am a molecular and cellular kind of person. So how do I look at these kinds of neurons? I have to have some way of purifying these Foss-expressing neurons. And this is a procedure that took a long while to develop. And actually, it's coincidentally enough, Danielle Gez was, well, anyway, she was born and brought up in Israel herself, now that I think about it, for the first 7 years. But anyways, she is one of those, she was one of those really persistent, overachieving MD, PhD type students. You know, it's not enough to do one degree or the other, you have to do all of them. And basically, she took a procedure, the idea for a procedure that many people had tried for many, many years. The idea of fluorescence-activated cell sorting has been around since the 60s, actually, yeah, mainly the 60s. And cancer people use it, immunology people use it. The reason we hadn't used it with adult brain tissue is that you need to make the cells single. You can't have clumps of cells. And so it requires a technique for separating the cells without blowing them up. And for a year and a half, and this is actually a teaching tool for people who start out in graduate school, but for a year and a half, we basically had DNA all over the slides. Okay, we just kept destroying the cells until we found the particular combination of enzymatic and mechanical methods to separate these particular neurons without blowing them up and making single cells. So it took a lot of diligent work. So once the cells are made single, they pass through a flow cell within this fax machine. And when they do, I should have said they're labeled with antibodies that are fluorescent. Okay? And you could actually label proteins that are inside the cell, not just on the outside of the cell. And when they do, a combination of lasers and photomultipliers will detect certain kinds of information. Forward and side scattering, but the other thing is they can detect the amount of fluorescence attached to these antibodies that it correlates with each cell. So if they have a lot of fluorescence that's correlated with Foss expression, then there's this fancy idea is that once they pass a certain position and it determines that this is a Foss expressing neuron, it tells basically the other end of that flow cell that add a charge to this particular cell, not the other ones. And then as it passes between two electrostatic deflection plates, it will guide that particular Foss expressing cell into one tube, whereas the remainder will go into the other tube. Okay, this is how we separate Foss positive and Foss negative neurons. Of course, we need the other antibody to tell you it's a neuron, because the vast majority of stuff that comes through, honestly, is debris. So you have to know that it's a neuron, and you need to label it whether it's got Foss or not. Once they're separated, it's just basic molecular biology. You can do whatever you want. Look at proteins here, or you can look at mRNA expression and a host of other things. Now, this next experiment I'm using, it's not even an experiment. Well, it's kind of an experiment. Basically, you take a rat, you inject it with methamphetamine, Oh, I didn't mention, that's another fun drug. And you're trying to strongly activate these particular neurons. And then you separate the Foss positive neurons from the majority of neurons that are Foss negative. And then we are trying to look at what kind of immediate early genes are induced in Foss expressing neurons versus the vast majority of neurons that are Foss negative. And when you use quantitative PCR to test this, you see the following. In the Foss positive neurons, in the dark bars here, compared in the methamphetamine injected animals compared to the saline injected animals, there's an increase in C-Foss mRNA expression and even more of Foss B expression going from saline to methamphetamine. I want you to notice the y-axis, always notice the y-axis, 0 to 40-fold. There's large increases in C-Foss mRNA and Foss B. And then the same goes for delta Foss B and then the other, but also in immediate early genes ARC, in this case Homer2, you see an increase in the Foss expressing neurons. What you do not see, at least not statistically significant, you do not get increases of these immediate early genes in the Foss negative neurons, even though they make up probably 99, well, they do make up 99% of those neurons. The vast majority of neurons do not increase their immediate early gene expression. It seems to be happening only in these Foss expressing neurons. For example, if we did the same experiment, but this time we just took homogenates of that whole brain area. Now look at the y-axis again, 0 to 4 instead of 0 to 40. You see increases in the methamphetamine injected animals of both C-Foss or Foss B, for example, but they're a lot smaller, 50%, 100%. They're not multiple fold increases. And here's the better example. For ARC and Homer2, you do not see an increase in the expression of these particular genes when you grind up the entire brain area. This is because those Foss negative neurons, that are the vast majority that are surrounding these Foss positive neurons, are diluting the immediate early gene effects that you're getting only in the Foss expressing neurons. In other words, these changes are happening only in the Foss expressing neurons. And we had pointed out in the other cases that these particular neurons are mediating that behavior. So people ask me, but we know neurons are turned on all the time. Tons of them turned on, and it's true. A lot of them are turned on if you stick an electrode in, do in vivo electrophysiology. Most of those neurons will be turned on at some point in time. What I think is happening, and what I know from Foss expression, but also from the results I'm seeing, what I believe is happening is that with increasing amounts of excitatory input, I said it properly at the time, but increased amount of input, there's an increased depolarization of the soma, increased influx of calcium. And then, but what I think is happening is that there needs to be a threshold amount of activation, a threshold amount of calcium influx to get activation of that MAP kinase that I was talking about, or other signaling mechanisms, to get this kind of a gene induction. And this is what I think separates the ensembles that some people would look at with in vivo electrophysiology, where many neurons are turned on, from the kinds of Foss expressing ensembles that I'm talking about, where 1% and often less have Foss expression within them. I think you need a certain persistent amount of input that's selected by cues and context to force these neurons to undergo some kind of molecular change. And I'll show you in a subsequent section that this also happens for electrophysiological changes. This, I'm just going to skip over this part. It's far too complicated. But basically, it was showing you that in many other cases, these are the other experiments we've done looking at using FACS to separate Foss positive, Foss negative neurons. But the idea is that we see the changes that we're interested in only in the Foss expressing neurons. And in a bunch of different brain areas. And again, we see them only in the neurons that were responsible for mediating that behavior.